Hi, everyone. This is Polar, and welcome to the first lecture on structural biology regarding energetics of molecular interactions. This is the same lecture that I gave live, just with a nicer recorded version for y'all to peruse on your own time. So, what is structural biology? Structural biology is really the study of how biological molecules are built. When we look at general biology that you may have taken in high school or undergraduate education, you've seen how molecules are formed. You understand how things are put together through dehydration synthesis or how things are broken through hydrolysis. And we focused a lot on the major macromolecules of life: nucleic acids, proteins, carbohydrates, and lipids. And these are all really important fundamentally on how they interact inside the cell and living systems. But that's really focusing on function and an overall general view. In structural biology and biophysics, we focus on molecular interactions and the forces that lead to synthesis of biomolecules. We understand how these interact in 3 dimensions. And that's really what structural biology tries to understand: the physics behind why the double helix is shaped the way they are, and why protein structures are folding the way they are into alpha helices and beta sheets. So this underpins a fundamental concept in biology and in really all of life: the idea that structure leads to function. If you understand the structure, you can understand the function. If you understand how the protein folds, you'll understand how that operates and fits in the cell. And we can also use this to understand pathogenesis: why some cells, if you change their structure, it suddenly leads to diseases and malfunctions in a body. A classic example would be sickle cell disease, where we have a different shape, the structure of the blood cell changes, and that has disastrous effects on the body. But before we go into those major and exciting topics, we'll look at the big themes of this course, and hopefully I'll be able to share more of my passion for structural biology. The first idea is energetics. Energetics is how molecules interact, and this is like the foundation to building our house of structural biology. We'll look at the central dogma and how things from genetic information is turned into proteins, which makes us and all living things. We'll also look at nucleic acid structure in depth and why the double helix is shaped their way they are to avoid repulsions and whatnot. And we'll also look at protein structures, understanding how they fold and how things govern, um, and how structure governs function. So energetics, we'll understand energetics, intermolecular interactions, and how this helps govern um, our 3 dimensional structure. And you may have encountered this in general chemistry. So we'll go over this in a bit more detail for those who haven't. We'll look at covalent and non-covalent interactions. Covalent interactions are very strong and are due to the sharing of electrons. We'll see this in the primary structure of macromolecules. For example, in DNA, we'll see that the backbone of DNA is connected through phosphodiester linkages. And these are covalent bonds between phosphate groups and, um, and the different backbones, the different components of the backbone. And we'll also see in protein structure how the peptide bond between amino acid residues are due to the sharing of bonds. And this rigid structure is the reason why some angles are just impossible for proteins. And these are all covalent interactions. And what's important about covalent interactions is that the breaking and forming of them is really what drives chemical reactions. And if you look at, and if you've taken organic chemistry, anything that partakes in the breaking of bonds and making of bonds is a reaction compared to the movement of electrons, which we saw more in resonance. But this isn't organic chemistry, so we'll keep this as these are very strong and that they help us understand the primary structure of molecules. What we're more interested in, at least at a structural biology standpoint, is the idea of non-covalent interactions. And these are typically weak. And many important biological interactions are non-covalent. An example from the Molecules of Life textbook would be conformational changes. We have this water molecule interacting with this side chain of an amino acid, and that just changes its conformation on the surface. It's not breaking any covalent bonds, so this is a non-covalent interaction. Or disruption of ion-ion interactions between these two oppositely charged amino acid side residues. All of this is due to breaking of non-covalent bonds, so no chemical reaction is occurring, just interactions between molecules or intermolecular forces. And these are non-covalent. So three types of interactions are really important to understanding structural biology, starting off with van der Waals interactions. You may have seen this as London dispersion forces or induced dipole, depending on your institution and how you like to phrase things. I personally enjoy using van der Waals interactions because it's fun to say. Van der Waals interactions, we could start by looking at a normal neutral atom with no charge. Because it has no charge, the electrons are in a cloud if we look at the electron cloud model. And mathematical analysis can really tell us, kind of map where things are, and we could look at orbitals and all of that. But for the sake of simplicity and looking at models, what we're interested in is what happens when you put this electron cloud near a charge, let's say a positive charge. This is where we create induced dipoles, where a temporarily mini magnet, as I like to simplify it as, is formed. And these induced dipoles create charges. And as we know from playing with magnets, opposites attract. And this creates a temporary or transient dipole. Just as a disclaimer for the rest of this, I'll be using a lot of general models and general analogies just to help this become more interesting and easier to digest for all levels of students. So it won't entirely be accurate, but this is the best way to encompass as many students as possible in the understanding. And these are general qualitative principles, and we'll hopefully be able to get into more quantitative principles later on in future lectures. So van der Waals interactions are denoted and kind of defined by weak interactions, weak attractions, and strong repulsions, meaning that individually their attraction's rather weak, but they have strong repulsion when they get too close. And we'll look at some Morse potential energy diagrams in a bit that explain this in more detail. What I want to denote, though, in van der Waals interactions is that the aggregate sum is very strong. For example, if we have geckos, which is a classic example, how they could climb up walls and glass. That's due to a strong aggregate sum of van der Waals interactions. So there's a lot of them that are acting on the toes of the gecko, and those can go up and down. So the aggregate sum is strong. Another interaction is ion-ion interactions, which, as it states, is due to a strong interaction in Coulombic electrostatics. If you remember from physics or from general chemistry, you know that Coulomb's law tells us the strength of attraction between two oppositely charged molecules based on their distance. And here we see the molecules of arginine and glutamate, which are two amino acids residues interacting due to their opposite charges. And these are strong interactions. So you need and we'll see this more and more in protein structure. Hydrogen bonding, on the other hand, is stronger than van der Waals, but weaker than ion-ion. And this is due to the polarization of covalent bonds. And we could use the Pauling scale for electronegativity to figure out partial negative and partial positives. How I like to see this is that electronegativity is like how much you want electrons, how much you want to pull electron density closer to you. And we'll see that oxygen is rather electronegative, meaning it will want to pull electrons toward it. So it's going to have this partial negative charge, exposing this hydrogen here with a partial positive. How I like to see this as an analogy is if you're sleeping and you have a blanket, the top of your body wants to be warmer than the bottom half, so it's pulling the blanket up. So similarly, if you think of the interaction, the oxygen being the top and your feet being a hydrogen, the oxygen will pull the electron density or the blanket closer to you, exposing kind of a positive charge on the hydrogen. And this is kind of the polarization of the bond. And hydrogen bonds are created by hydrogen bond donors and hydrogen bond acceptors. It is important to note that hydrogen bonds are not between two hydrogen atoms. It is between an electronegative atom and a hydrogen bonded to a hydrogen and another electronegative atom. And this is the hydrogen bond, and due to depolarization. Because if we had two hydrogens, then you don't really have that electrostatic force. You don't have an attraction between a partial positive and a partial negative. And these arrows denote the direction of electron flow. In energetics, like I said before, this is a Morse potential energy diagram that shows a generic comparison between van der Waals, ion pairs, and hydrogen bonds. We have potential energy in kilojoules per mole on the y-axis, and on the x-axis, we have radius measured by angstroms. And this shows the potential energy depending on how close two molecules are. If we look to the far right, or as the radius gets bigger and bigger, we'll see that the energy is rather close to zero because they're so far apart, there's no attractive force to pull them together. Likewise, if we go to the far left, we'll see that the energy spikes significantly due to strong repulsion between two atoms by getting too close. So this brings us to the idea of optimal distance. Well, atoms, if we want to think of 3D structure, we don't want them too far, otherwise it's not stable, and we don't want them too close. It's also not stable. It's pushing too far apart. So this optimal distance is the sum of their radii at the lowest energy point. And again, like I said, this is a qualitative analysis. The radii are different when we look at van der Waals, ions, and hydrogen bonding, and I'll go into that in future lectures. But the optimal distance is where the energy point is the lowest. It's where there isn't going to be more attraction and there isn't going to be more repulsion. It's this bottom of the well. And how I like to think about it is using a marble analogy. If you drop a marble on the left side, it will roll down to the bottom of the peak because it's going from high energy to low energy. It wants to be in the stabilized portion. Similarly, if they're on the right side, it'll roll down because of attractive force. So all atoms, when they interact, want to be in the most stable and low energy. So they'll try to achieve this optimal distance at the bottom. And we can calculate those in quantitative analysis. And the stabilization energy is the difference between zero and lowest. And this is important when we talk about thermal energy here. So thermal energy is the energy of the system based on the movement and vibrations of molecules. We could kind of define it as how much energy is transferred from molecule to molecule when they collide. And at thermal energy, at room temperature, if we put it at 300 Kelvin for simplicity's sake, is approximately 2.5 kilojoules per mole, which means that's as much energy that's going to be transferred when they collide with each other. And you could figure this out using a Boltzmann factor and temperature or the gas constant and temperature. And that I won't be going into. But what's important is that this thermal energy tells us whether an interaction could be disrupted or not. If we take, for example, at room temperature and look at this ion pair has a stabilization energy of approximately 20 kilojoules per mole. It's massive, which means that we would not expect thermal energy and random collisions to break an ion pair at room temperature because the thermal energy is less than the stabilization energy. But if we look at this van der Waals interaction, which if we're looking at it is approximately like 1 kilojoule per mole, we can expect that since it's less than thermal energy, then random collisions could break the van der Waals interaction. And this concept underlies why high temperature is required to separate DNA strands. Because of the hydrogen bond pairing, you want to have enough so that it disrupts that bonding, and why proteins have an ideal temperature range before denaturing. And this is very important when we look at that later. So, like I said before, there's strong attraction depending on your interaction. And this attraction, as the radius gets large, the potential energy gets closer to zero. It's like pulling two magnets far apart. At some point, they're going to stop wanting to come together. Similarly, with when they get closer, the attractive force gets stronger and those two magnets will come together. And I'm using magnets just as an analogy here. It's not entirely accurate to what's happening, but for simplicity's sake, that's how they are when they're coming too close. This transient, whether it's a transient dipole due to van der Waals, hydrogen bonding, or ion pairs. Ion pairs are basically like magnets with opposite charges. Repulsion. An example of this, and I'll be careful, is that atoms act as hard spheres. And for structural biologists, this is a good way to just generally and qualitatively understanding what that this idea of repulsion, meaning that when they get too close, they collide too much, and the electron clouds try to repel each other. And you could see that up here on the left when the radii are way too close. Now, this is just an analogy. And for structural biologists, this is somewhat good enough to think of it as hard spheres that they get too close and repel. If you look into more chemistry, you'll understand that there's more ways we can actually calculate it and explain it. But for the sake of this course, we'll understand that when they get too close, van der Waals repulsion or any kind of repulsion will push them apart. And this is really strong repulsion, is when they get way too close. And why is this important? This is a diagram of DNA from a pi-mole session. And you'll see that this is really important to understanding what stabilizes DNA. We'll see that hydrogen bonding between base pairs is stabilizing, but it's not the driving force of DNA structure. That's because in an aqueous solution, meaning water, the hydrogen bonds are always competing with water molecules. So that doesn't make it as strong as it could be. This also explains why we don't have two purines or two pyrimidines interacting with each other due to the importance of distance between hydrogen bonds. And this explains Watson-Crick base pairing that we'll go into later. So hydrogen bonding is explained through these interactions, and we'll go into quantitative analysis of distance later. Base stacking is really important, and that's the driving force of creating the DNA double helix. And this relies on the van der Waals forces and Coulombic electrostatic interactions. The dipoles that are created through this base stacking help stabilize the double helix. And as we said before, the aggregate sum is really strong. So that's why base stacking is important. And we'll see that sugar conformation is another thing we can explain when comparing A-form DNA, B-form, and Z-form DNA. The sugar conformation, or the sugar pucker, is explained because in a heterocyclic ring, these atoms don't want to be in a plane. And that's kind of hard to denote here, but I'll show a better diagram in a future lecture. But the reason why it's in this ring and in 3 dimensions, one of them will pop out or pop below the plane. And that all depends on what's there, whether it's a hydroxyl group in RNA or just a hydrogen, and it's avoiding the repulsion of the phosphate backbone. And all of this is explained through intermolecular forces. So this 3D structure, while the primary structure is denoted through these covalent bonds, the 3D structure is largely governed by these non-covalent interactions. And the spacing and optimal distance explains why there are 10 turns, 10 base pairs per turn of DNA, why each of them takes 36-degree rotations, and why the double helix is right-handed in B-form DNA versus left-handed in A-form DNA, or left-handed, sorry, in Z-form DNA, not A-form DNA. But these fundamental concepts explain why they are shaped the way they do. So that's it for energetics. And these principles are really important for our understanding of what happens. So feel free to reach out to me here. Just some credits, textbook credits to the Molecules of Life textbook by Kurian, Boyana, and Weimer for their fantastic diagrams, and credits to the template for this presentation. So feel free to reach out. I'm more than happy to answer more questions about this topic.